Thank you. Thank you very much for joining us and accepting our request. This is the fifth lecture of our lecture series, and I would like to, welcome you, warm welcome here in the series of lectures here. So Dr. Amit Sharma, who is the director in MR, is also joined. Where I guess you are meeting him just before this lecture. So, if you allow, can we start now?
Sorry, Sachin, would you like me to start now, or?
Okay, so before we start, I would like to read some lines about Professor Dominic. So Professor Dominic holds a joint appointment as an MRC clinical research professor at Oxford University and has a head of malaria program at Wellcome Sanger Institute, UK. He is the director of MRC Center for Genomics and Global Health, which is a joint research program of Oxford University at Sanger Institute. He trained in clinical pediatrics. He started research on cytokines in Charles' lab in Boston in 1985, and following year he went to Gambia to study molecular mechanism of malaria fever. In 1989, he moved to Oxford University, maintaining a clinical research program in the Gambia. His group started working on genetics around 1994 as a way of getting the basic research question of malaria pathogenesis. In 2000, he moved to Wellcome Center for Human Genetics in Oxford, and in 2005, he took up a joint appointment at Wellcome Sanger Institute. The overreaching goal of his current research is to help practical problem in global health by using new approaches in genomics, epidemiology, and statistics. In 2005, he founded a data sharing network for Malaria Genomics Epidemiology Network, which is Malaria Gen, which is very popular now, which has fostered reproductive research collaboration in more than 40 malaria endemic countries and has become a model for equitable sharing of genetic data and research capacity building in resource-poor settings. So with this and without any further delay, I would request Professor Dominic to start his talk.
Thank you very much, Sachin. Before I start, can I just check, can you hear me okay, and can you see my slides?
Yes, we can hear you and we can see your slides as well.
Well, thank you, Sachin, and colleagues for inviting me to speak in the NIMR and MERA-India lecture series. It's a great honor. Much of the work of our team at Sanger is focused on partnering with researchers in malaria control programs around the world to use genomic surveillance as a tool for malaria control and elimination. And as you said in the introduction, Sachin, we do that through a data sharing network called Malaria Gen, which has partners in over 40 malaria endemic countries. With new technologies, there are extraordinary opportunities to use genomic surveillance to tackle the spread of parasite resistance to artemisinin and combination therapies, for example, as it's happening in Southeast Asia, and also to tackle the spread of mosquito resistance to pyrethroid-treated bed nets, for example, as we're seeing in Africa. And I'll say a bit more about Malaria Gen and the practicalities of genomic surveillance later in the talk. But today, I want to actually share with you a more basic biological discovery, an exciting discovery that we've recently made concerning host-parasite interactions in malaria. To give you the, now let me just get the, I need to find out how to advance my slides, excuse me. So, humans have genetic factors that affect their resistance to malaria. The classic example is hemoglobin S, which encodes for sickle hemoglobin. It's caused by a single nucleotide polymorphism that causes a change in the beta globin chain. And it's a classic example of balancing selection, because individuals who are heterozygous for the hemoglobin S variant are protected against severe malaria, whereas those who are homozygotes get sickle cell disease. And if we look at the global frequency of hemoglobin S, the allele frequency, it very much correlates with the historical endemicity of malaria, of falciparum malaria. About 20 years ago, we embarked on a study to endeavour to discover novel host genetic factors that confer resistance to severe falciparum malaria. We work with partners in 11 countries, 9 in Africa, but also Papua New Guinea and in Vietnam, to collect a total of about 16,000 cases of severe malaria with about 22,000 population controls. And in those samples, we genotyped millions of SNPs and ended up examining over 10 million variants in the genome and asking, were they associated with susceptibility to severe malaria? And we've published a number of papers about that. Some are listed at the bottom, but in brief, in 2019 in Nature Communications, we provide a sort of summary of what we've found, and it's summarized in this graph. And what we're seeing on this graph are millions of points, about representing over 10 million variants in the human genome. The numbers at the bottom are the chromosomes that they're on. And what we're seeing on the vertical axis is a measure of statistical significance for an association of that locus in the genome with resistance or susceptibility to severe malaria. And you can see there's a number of quite strong signals. There's not hundreds of strong signals, but there are a number of well-defined signals, and I'll talk a bit more about those in a minute. Well, let's go on to that then. So if we look at those top signals that we see in the human genome, what are the most prominent forms of host genetic resistance to severe malaria? We find those all localize in variants of the human red blood cell. And the top one is indeed hemoglobin S. That gives a relative risk of approximately 0.1 for severe malaria. In other words, there's about 90% if you're carrying a copy of the hemoglobin S variant, you're about 90% protected against developing severe malaria, which is pretty good. It's as good as having a COVID vaccine for COVID, at least as good as that. A major discovery that came out about 3 or 4 years ago was another blood group variant called DAN2, which is a structural variant of the glycophorin genes that affects a receptor for the parasite that gets into the red blood cell, and that gives a relative risk of 0.6 for severe malaria. Another discovery that came out of early GWAS studies was a variant of the calcium channel, the ATP2B4 calcium channel on the red cell. And it turns out that blood group O, an extremely common variant globally, also gives some protection against severe malaria. And these are the top signals we've seen so far. Now those loci, they're strong signals, but they account, but what we can do is work out from genomic data how much of a heritability of severe malaria they account for. And our data suggests they only account for about 10% of the total heritability. So there's a lot of stuff that we still haven't discovered despite this large study. We know there are a number of loci that are well known, and they do have an effect on malaria susceptibility, but their effects are relatively weak. They're statistically significant, but they're relatively modest effects, and so they don't show up in this large genome-wide association study because the p-values in a genome-wide study don't reach statistical significance, although we know these are true associations. Our genome-wide association study has revealed a lot of other candidate loci that might be involved, hundreds, but they've yet to be validated. So we're still left with a lot of stuff that is yet to be explained. Now, a key question out of this is it just about human genetics, or are there parasite factors that affect the clinical outcome of malaria? And that's a big question. It has many different elements to it, but there's a very specific version of that question, which is, have parasites adapted genetically to avoid the protective effect of host genetic resistance factors? So what's going on in the parasite genome? So the study I want to tell you about is a recent study that was led by Gavin Band and Kurt Rockett at the Wellcome Sanger for Human Genetics in Oxford, with working with the Sanger Institute to sequence a lot of parasite genomes, which I'll talk about in a minute, and to analyze the data to see if we could find an association between human and parasite genetic variants. I'll talk a bit more about that in a minute, but let me say that this particular study was focused on data that we had collected over the years from the Gambia, led by Muminatu Jallow at MRC the Gambia, and in Kilifi, Kenya, led by Tom Williams there. So a collaboration between Gambia, Kenya, Oxford and Sanger. But building on things we discovered previously with our colleagues in the Malaria network. So this is what we did. We took some of the severe malaria cases from the previous human genetic association study, about 2,500 from the Gambia, 2,500 from the Kenya, and in those samples we sequenced the parasite genome. Now that was, when we started out doing it, it was non-trivial because of course most of the DNA in our blood samples is human DNA, not parasite DNA. So we used a number of different strategies to sequence the parasite genome. One was to sequence all the DNA in there, both human and parasite, at very high depth. So very high depth sequencing of everything that was in the sample. And another strategy we used was so-called selective whole genome amplification of the parasite genome, where we go in and enrich for the parasite DNA and sequence that. So we did that, and we, having sequenced these over 5,000 cases, we did variant calling and quality control. And then we merged that with the data set that we'd previously generated that I talked about, the human genotypes that we had from our previous study of human genetic resistance to malaria. And that led to a data set that had an overlap between parasite genomic data and human genomic data of almost 3,500 samples. Sorry, I need to go back now. Okay, so the next thing that Gavin and colleagues did was to test for pairwise association between human and parasite genetic variants using a logistic regression framework and some software that Gavin developed, which is available online. In the parasite genome, we focused on biallelic variants, variants that just have two different alleles, typically single nucleotide polymorphisms, biallelic variants across the genome. We excluded the very complex parts of the genome, the subtelomeric regions, and we included loci that were multi-allelic, and we insisted that variants be seen at least 25 samples. So that left us with 50,000 variants that we analyzed in the parasite genome. And in the human genome, we focused on the known malaria resistance alleles, some of which I've talked about, a bunch of several thousand other loci, including variants in blood cells, HLA alleles, and other putative genetic associations with malaria. And out of this, what we discovered was that, this is a complex figure, but I'll give you the bottom line. We discovered 3 positions in the parasite genome that were strongly associated with human genetic variants. And in all of those 3 loci, the main thing they were associated with was the HBS genotype of host. So let me just walk you through what this complex diagram shows. It shows on the top panel a view of the parasite genome. And what we're here is seeing signals of association in the parasite genome, positions in the parasite genome where the parasite genotype is associated with some variation in the human genome. And there are 3 major signals, 2 on chromosome 2, 1 on chromosome 11. I'll talk about a bit more. And what we're seeing on the bottom panel is what's going on in terms of our human genetic analysis. Here we're not analyzing the whole human genome. What we're analyzing is a whole bunch on the left-hand side, a whole bunch of human genetic variants that we believe to have some evidence of association with malaria. And you can see the one that comes out most strongly as being associated with parasite variants is this RS334 variant, which is the haemoglobin S mutation. And then over here on the right-hand side, we see a whole bunch of other genes that we tested of interest because they were blood group genes. So 3 big signals of association. So let's look at those in more detail. There's a big signal on chromosome 2 here, another one on chromosome 2 about 200 kilobases away, and another on a completely different chromosome, chromosome 11. We call these pfsa loci, meaning Plasmodium falciparum sickle associated loci. Let me spell that out in more detail. In other words, these are positions in the malaria parasite genome where there's a variant, and that variant is associated with sickle. In other words, if someone is carrying sickle, they're more likely to have parasites of that genetic type. And we call them pfsa 1 plus and 1 minus. If it's pfsa 1 plus, it means it's carrying the variant that is more likely to be carried by people with sickle. And if it's carrying the pfsa 1 minus, it means they're carrying the variant that's most likely carried by people that don't have sickle. So we found these three positions in the genome. Now, when we analyze the protective effect of severe malaria, and we can do that by a relative risk analysis and say, okay, what if someone was carrying one of these parasites? Would sickle still protect them against malaria? And we find that if the person is carrying variants that parasites with these pfsa variants, it greatly reduces the protective effect of sickle against severe malaria. In fact, I talked about 3 loci. It turns out that if someone has a parasite that has the variant at all 3 loci, then they've got hardly any protection from sickle. The protective effect of sickle is down to about 20%. Whereas if they've got carrying parasites that have none of these loci, the protective effect of sickle is enhanced. It goes up from about 90% to over 98%. So what we've done on the right-hand side is we've changed our estimate of the protective effect of sickle. What we're seeing on the top bar is there's a very strong protective, an extremely strong protective effect, much stronger than we thought previously, if the parasites don't have these variants. But if they do have these variants, then the protective effect is very much less. In fact, it's barely there. So that raises the question, well, what do these mutations do? And the answer is we don't know. At the pfsa1 locus on chromosome 2, the signal of association, the genetic signal of association that we see, includes non-synonymous changes in a gene that encodes an SL-CoA synthetase, so that might be a clue. At the pfsa3 locus on chromosome 11, we don't know what's going on, but fascinatingly, a recent study from Mali by Joseph Salins, Mohamedou Diakité and Steve Taylor and colleagues found that one of the genes of that locus that I've listed here is amongst the most differentially overexpressed genes in children who are carrying sickle. In other words, if you're sickle and you're carrying parasite infection, and we do a transcriptome analysis, we find that genes at this locus are very overexpressed if you're carrying sickle. And further analysis of this data set in collaboration with Steve Taylor and colleagues has shown that the variants of this pfsa3 locus appear to regulate the expression of that gene and appear to be linked to a neighboring copy number variant. So on this slide, all I've shown you is that we've got a number of clues that might help us to do some molecular dissection of what's actually going on here. But at this point, what we can say is we've got an extremely strong epidemiological association, which undoubtedly is telling us something. We don't know mechanistically how these loci are affecting the sickle protective effect. Now, I'm going to divert briefly onto a slightly different topic, but I'm going to come back to our discussion of the pfsa alleles and sickle in just a minute. But I want to divert because I want to tell you about another major project we do, one of the biggest areas of our work, which is working with partners around the world to have what we call a Malaria Parasite Observatory and a Malaria Vector Observatory. Here I'm talking about P. falciparum, but we also have similar things for Plasmodium vivax and for Anopheles gambiae, Anopheles funestus. And our goal has been to work with partners around the world to assemble an open resource of data on genome variation in malaria parasites and malaria vectors. And we have a model for doing that. We work with our partners either to sequence the samples or to help them analyze their own data. And our model is that we have try to have an equitable sharing network whereby if partners contribute samples, they get the data back immediately, and we support them to analyze data on those samples for their own purposes. And then after they've done that, we share it more generally. So we're trying to set up a model where open data sharing can be aligned with equitable data sharing and benefit to partners in malaria endemic countries who wish to analyze their own data and generally their own data. So, and the relevance of this to the PFSA problem will become apparent in 1 minute. But let me just simply say that the current release of our Parasite Observatory data, the PF6 data release, has 7,000 Plasmodium falciparum samples from 28 malaria endemic countries. And the data set includes high quality genotype calls on over 3 million variants. On the right-hand side, you can see where it's published in Wellcome Open Research. You can see that we publish it in an alphabetical author list, like theoretical physicists, like particle physicists do. And you can access all these data if you go to the Malariagen website, malariagen.net/parasite. You can get access to these data through both data downloads, but also a web app we've developed to help browse the data. And we're currently working to release the next version, PF7, which will have high quality data on over 16,000 samples from over 30 malaria endemic countries. That's coming very shortly. And we hope by next year, and that PF8 release, to have doubled that again. So the size is currently growing exponentially. Now, the reason for going on to that is that this provides an extraordinary repository to ask all sorts of questions. The sorts of questions that interest us most in this Parasite Observatory is how we deal with things like the spread of ACT resistance, multi-drug resistance, the questions like whether parasites have HRP deletions that affect rapid diagnostic tests, and other practical matters in malaria control. That's really our driving force. But it does provide also a great scientific repository to ask more basic biological questions. So now let's come back to the PFSA variants and look in the PF6 dataset and ask, what is the distribution of these variants? Because thus far I've only told you about data from the Gambia and Kenya. And what we can see here, sorry, it's rather small writing, is that if we look across the different countries that are contributing data to the PF6 data resource, we essentially find over on the sort of more left side of the first panel, what we're seeing is a relatively high frequency of these variants in Africa. And what we're seeing more on the right-hand side of this first panel is an almost complete absence, or actually a complete absence of these PFSA variants in Southeast Asia, in Bangladesh, in Indonesia, Papua New Guinea, South America. Well, the South America data are a little less clear. And what we can see on the right-hand side is that if we look just in Africa alone, we can see that there's a rough correlation of the frequency of these PFSA variants with the frequency of sickle. So in areas where the sickle frequency, allele frequency, is about 10%, the frequency of the PFSA variant in Cameroon goes up to over 50, goes up to around 50%. So in other words, the geographic distribution of these variants is consistent with the distribution of hemoglobin S in the human population. Now, the next thing that we find is that it is to do with the correlation between these three alleles. There's a term in genetics known as linkage disequilibrium, which refers to whether two alleles are correlated. In other words, if I type a bunch of samples in a particular location, and if I know what genotype they have, at a particular variant at this position in the genome, can I predict what genotype they'll have at another position in the genome? That correlation is known as linkage disequilibrium. In the case of malaria parasites, linkage disequilibrium is normally extremely weak because sexual recombination of the parasite as it passes through the malaria vector leads to a jumbling up of loci. And we can see on the right-hand side in the pf6 data set that in general, on average in the parasite genome, there's very, very poor correlation between alleles if they're separated by more than a kilobase, more than 1,000 bases. Now, in the case of these pfsa alleles, they're separated by much longer than that. On the pfsa 1 and 2 are separated by about 200 kilobases, and pfsa 3 is on an entirely different chromosome. So we'd expect them to be very little weakly correlated. Yet, when we look at their correlation in our study, in the study I just talked to you about, and in the pf6 dataset, they have actually an extremely strong correlation coefficient. They're in very strong linkage disequilibrium. And in this slightly complex diagram on the right-hand side, what we can see is if we plot their correlation versus the average correlation of loci in the genome, it's way an outlier. Here's the correlation, for example, between pfsa1 and pfsa3 in Kenya. It's an extremely strong outlier of the normal pattern. So we're left with many questions. What's the biological function by which these pfsa variants enable parasites to evade the protective effect of HBS? Why haven't, what maintains these variants in strong linkages to disequilibrium when all of the other loci in the same parasites in the same genome have very weak linkage disequilibrium? And then another question is, why haven't these variants that we're seeing gone to fixation? In other words, why they aren't seen in all parasites? If it benefits the parasite to if it allows the parasite to evade sickle, why don't all parasites carry these particular variants? Now, one can hypothesize that that last question is some form of balancing selection in the parasite, that just as with the human, it doesn't benefit human populations for everyone to be carrying HBS. And in the parasite population, it may be that the parasite population needs to maintain that diversity because there's some appropriate balance between the benefits for the parasite and the risks for the parasite in having these pfsa alleles. So understanding the basis of that potential balancing selection is one of the key questions for the future. Going back right to the beginning of this talk about what are the human genetic variants that protect us against malaria, and as I mentioned, we've so far only found the tip of the iceberg. How are we going to start finding all the other variants? One possibility is there's just thousands of other very weak effects. We've pulled out, I've told you about the big blockbuster effects for protection against malaria, but there might be many, many much weaker ones. And that's an entirely plausible hypothesis. Another plausible hypothesis is that it requires combinations of different variants to protect. But another important thing that arises from this discovery I'm telling you about is that a lot may be revealed when we understand more about host-parasite interactions. So my working hypothesis, just a personal view, is that when we're able to analyze more complex forms of parasite genetic variation, which at the moment are very difficult to analyze, such as the MERA-India invasion genes and the VAR genes, they have complex forms of genomic variation. My hypothesis is that we'll discover many more host-parasite genetic interactions of the sort I've just been telling you about today. And my second part of that hypothesis is that by discovering those host-parasite genetic interactions, we'll then get a better understanding of both the host and parasite genetic determinants that affect the clinical outcome of malaria. So I think it's a big area for the future. Now that was all about basic science, but I want to now just come back to the massively important question just right at the end of the talk of how, as a malaria community, researchers, control programs, policymakers, funders, governments, how are we going to create the genetic surveillance resources that we need to support the control and elimination of malaria? You know, COVID, the pandemic has taught us many things, and one has been the extraordinary importance and value of genomic surveillance in tackling the disease. Knowledge of the alpha variant, the beta variant, the delta variant, the next variant that's coming down the line, has helped us hugely in understanding the epidemiology of the disease, and in the case of the UK, has been instrumental in informing national policy about lockdown, vaccine purchases, vaccine strategies, and so on. Malaria parasites and vectors are much more complex than SARS-CoV-2 viruses, but the same principles apply. How are we going to now start to use these genomic surveillance tools to better control the spread of antimalarial drug resistance, the spread of insecticide resistance, and to target our interventions for maximum impact? And there's many different parts to that problem. One small part of that problem, but small but important part, is creating systems for equitable data sharing, and when all parties feel comfortable with the arrangement and all of the appropriate regulations have been satisfied, then where appropriate, sharing the data openly. And in Malaria Gen, we are working to create open data resources. I told you a bit about our PF6 data resource, and I encourage you to visit the Malaria Gen website to explore some of the apps and data we've got on that. For those of you who are interested in the vector, I strongly encourage you to look for to look at our section on the parasite and go to our Malaria Gen vector data user guide, which for those of you who are geeks and into Jupyter notebooks and Python, will give you an extraordinary range of tools and resources to analyze this rich data set of African Anopheles parasite and Anopheles gambiae mosquito genome variation. There's a wonderful set of tools to look at population structure, insecticide resistance, and other features in that data set. Currently, our work on mosquitoes is focused very much on Africa, but we are now starting to do some work on Southeast Asian vectors, and of course we would absolutely love to discuss the possibility of collaborating on the vectors on the Indian subcontinent. And a crucial part of our mission going forward, beyond the basic discovery science I've been talking with you about, is to enable different countries to share genomic surveillance data on parasites and mosquitoes with governance structures and well-curated data resources. We want that genetic surveillance data to be translated into actionable information tailored for specific use cases in malaria control and elimination. Our team, Sanger, has been very closely involved with the UK national response to COVID, and a lot of our work has, over the last 18 months, has been learning how do you work with government to get them the sort of data that they need to make their decisions. And it took us a long time to learn how to do that, but now we're into it and we work on a daily basis with the UK government on surveillance data. And we also want malaria control programs to have access to services and technical support to collect genetic surveillance and to use it in their decision making. So on that note, I will finish and just say greetings to our Malaria Gen partners around the world, and a greeting to all of our colleagues in India from our team at Sanger and from our team at Oxford. So thank you very much indeed for the invitation to this talk. I'll leave it there. Thank you.
If you so we can have some questions here. Can we take some questions here?
I'd love to.
Okay. So the question is, why the Southeast Asian countries' data did not include India while associating pfsa variant and HPS? Second part of the question is, further, did you try to associate submicroscopic and low density PF infections and their pfsa variant with HPS?
Well, let me take the first of those questions. Why don't we have data from India? Well, it's just a sad fact that thus far our Malaria network hasn't included Indian Plasmodium falciparum samples. We are looking forward to changing that, and actually we're having some very interesting discussions about how we can partner with NIMR about somehow including data being generated in India into that database. So we would love to include that. It's one of the big missing things in our data set, and I think in the global data set. So that's something that we are extremely highly motivated to work on. Interestingly, we do have a better representation of Indian samples in work we're doing on Plasmodium vivax through a Plasmodium vivax network led by Sarah Alburn in Australia, and so that will be coming out in the near future. But that's definitely something that for us is one of the big goals of the next year, to improve our global representation in general, but most importantly to establish a strong basis of partnership in this area with India. So that was the first one. Could you just repeat the second one, because I'm not sure I fully got the second question.
So another question is, did you try to associate submicroscopic and low density PF interactions and their pfsa variant with HPS?
Okay, that's another great question. On that second one, it's very perceptive, because what I've talked to you entirely about samples of severe malaria, because they just happen to be ones that we were able to do this analysis on. I can tell you that there is work going on to look at this in more general, just uncomplicated malaria infections. This is not quite what the question is about, but there is work going on to look at uncomplicated malaria. And I can tell you, in preliminary data, the association holds very strongly there as well. So this is not just confined to severe malaria. Essentially, whether an individual has mild malaria or severe malaria, if they're carrying sickle, they're more likely to have this pfsa variance. We're quite sure about that now, but more work is needed in that area. Now, the question is, what happens about someone who's just carrying malaria parasites but has no symptoms? Does that also enrich pfsa? That's an absolutely fascinating question. I mean, I'm going to bet that there is an association, but until we've done that, we don't know. And setting up that study is very important. And I know people are thinking about it, but certainly if anyone on this in this lecture is interested in that question, I think it's a very important one. Why is it so biologically important? Because what is at the root of that question is, okay, what causes someone with sickle to have a higher propensity to carry parasites of a particular genotype? I mean, presumably the mosquitoes are pretty indiscriminate. I imagine that the mosquitoes are just infecting everyone with everything at random. That's just a guess. So presumably everyone is acquiring parasites of every genotype, but at some point in the process, parasites of this genotype win out in individuals who are carrying hemoglobin S. And I think what underlies this question here, why it's so interesting, is at what point does that enrichment of this genotype happen? Does it happen as soon as the parasite's getting into the host, or does it happen as they're starting to get symptomatic, or is it so I think it's a really basic question. And then the last question is, do we have data on severe vivax and HPS? Well, I'm afraid we haven't had the chance to work on severe vivax, and again, that's a fascinating question. I would love to have more data on that. We don't have data on severe vivax and HPS.
Okay. Another question is, what's your perception of other hemoglobinopathies like thalassemia and malaria protection?
Well, alpha thalassemia undoubtedly does have a protective effect against falciparum malaria, and Tom Williams in Kenya has been doing wonderful work to dissect that out. And with Kirk Rockett and colleagues in other malaria endemic countries in Africa, there is now a body of work to try to really lock down what is the protective effect of alpha thalassemia on a more comprehensive level. So there is a protective effect, but it's not nearly as strong as sickle. G6PD deficiency is another fascinating example. We've done a lot of work on that, and it turned out in our analysis of G6PD deficiency that it has a protective effect against cerebral malaria, a modest one, but it actually makes severe malarial anemia worse. So when we look at severe malaria as a whole, which is the combination of cerebral malaria plus severe malarial anemia, with G6PD deficiency, it works out more or less even because it's making part of the phenotype worse and part of the phenotype better. So there are undoubtedly quite complex things going on with other hemoglobinopathies, and we really haven't got to the bottom of all of them. And of course, one of the questions raised from this recent discovery is, would these things be clearer to us if we knew the parasite genotype? Because it's clear in the case of sickle that knowing the parasite genotype, we can actually get much stronger effects of sickle if we are correct for the parasite genotype.
Okay. The second question is, did you find role of other malaria protection variants like ATBB 2B4 at Gambia and Kenya protection against severe malaria together with sickle protection?
I think the question there is there an interaction of these things? So ATP2 clearly protects against severe malaria, sickle clearly protects severe malaria. I think the question is, do they interact together? That is actually covered in our paper in Nature Communications in 2019, where Gavin Band and colleagues have looked in detail at the additive effect of different malaria protective loci and asked, are they additive effects? Are they synergistic? Are they epistatic? And the bottom line is they sort of end up adding up in a fairly linear way. There's not evidence of big epistasis going on. So the answer is basically there's a protective effect of sickle, there's a protective effect of ATP2B4, and if you've got both, you essentially just add those two protective effects to first approximation.
Okay, I think this is the last question, I guess. Have you tried to link pfsa variants and drug resistance marker or PFHRP2 deletions?
Well, I think so. Let me think about how best to answer that. We certainly in our scans of linkage disequilibrium asking, what I've told you is that if you're carrying a PFSA, if a parasite is carrying PFSA1, it's much more likely than average to be carrying PFSA3. So the question is, I think the question that's being asked here is, if I'm carrying a PFSA1, am I more likely to be drug resistant? Am I more likely to have a PFHRP2 deletion? The second, on the issue of drug resistance, we haven't found any evidence of that. The LD, as far as we can tell, at a very high level, it does not affect the drug resistance loci. But we haven't looked at that in detail, and we certainly haven't looked at the HRP2 deletions in detail. So our current assumption is these things are not linked, but we haven't examined that in incredible detail.
Okay, so one more question. Is there any observed increase in PFSA frequency over time?
No, we've looked at, we've done some initial analysis of temporal trends, and there's sort of variation that looks fairly random, and it looks like sampling, it's probably largely due to the confounding effects and noise of sampling. So we don't see any evidence of a consistent trend. Our hypothesis is that these must have been around for a very long time. I can't give you concrete evidence of that, but all the data would suggest to me they have probably been around for thousands of years, just as sickle has. But that's just a hypothesis rather than a statement.
Okay, so with this, I think we can stop here, and I would like to thank you very much from the entire team of NAMR and from MERA-India that you were kind enough to accept our invitation and to spend your precious time with us. And also, I would like to invite you whenever the pandemic situation is better, you come here and we would like to have your talk in person at NMR.
Thank you very much.
It's
been a great delight.
Yeah. With this, I would end this session here, and thank you very much again.
Thank you. Goodbye.
Thank you. Bye-bye.